Welcome, everybody. This is a really pleasure to welcome the director of the NIH, Dr. Monica Bertagnolli. She is the 17th director of the NIH and took office just about a year ago in November of 2023. She was the first surgeon and the second woman to hold this role, and she transitioned seamlessly from her appointment as the 16th director of the National Cancer Institute. So prior to her federal service, Dr. Bertagnolli held appointments as the Richard E. Wilson Professor of Surgery and Surgical Oncology at Harvard Medical School, surgeon at Brigham and Women's Hospital, and a member of the Gastrointestinal Cancer Treatment and Sarcoma Centers at Dana-Farber Cancer Institute. As a physician scientist, Dr. Bertagnolli has championed initiatives to ensure the translation of foundational research into clinical applications, a vision she continues to bring to the fore as NIH director. She's a past president and chair of the board of directors of the American Society of Clinical Oncology, and previously served on the board of directors of the American Cancer Society and the Prevent Cancer Foundation. She was elected to the National Academy of Medicine in 2021, and we're delighted to have Dr. Bertagnolli join us today to discuss her priorities for NIH and the role of basic biomedical research within the vision. This presentation is being recorded and will be posted along with Dr. Bertagnolli's slide presentation to the FASEB volunteer portal later this week. We'll have a moderated Q&A following the presentation. So Dr. Bertagnolli, thank you so much for coming, and I'm just going to turn it over to you.
Oh, thank you so much. Thank you so much, Dr. Garvey, for that kind introduction. And thank you all for the opportunity to speak to you today. FASEB advances health and well-being by promoting research and education in biological and biomedical sciences. And as a result, its many member societies are critical NIH partners. We're grateful for this relationship, as our goals are very much aligned. Today, as a relatively recently appointed NIH director, I'd like to provide some comments on the importance of fundamental research, an aspect of biological and biomedical science that is so steadfastly championed by FASEB. Next slide, please. So today I'm going to briefly describe one of my own areas of research interest, make the case for more cross-disciplinary collaborations based on heuristic models of disease. I'll give a specific example prominently in the public eye today that starkly illustrates how successful disease prevention and treatment requires insights derived from fundamental research. And I'll also briefly describe NIH's funding for basic science and end with some guiding principles for our work. Next slide, please. First, seeking a fundamental understanding of health and disease. Next slide. I've spent my career studying cancer. As a clinician scientist, this means close ties to both the laboratory and the clinic. A particular area of interest for me has long been the role of inflammation in early tumor formation. And as a surgeon, I've always been intrigued by how a critical feature required for human health, the ability to heal a wound, engages a tightly coordinated inflammatory response in a process that the only time outside of a developing human being controls symmetrical stem cell division. And as a cancer researcher, I've been very interested in understanding how this essential program that's so nicely regulated during normal wound healing can be corrupted in ways that lead to the uncontrolled growth that initiates and maintains a tumor. Next slide. A much more comprehensive view than this is a heuristic model of cancer that I've often been drawn to, presented by Dr. Doug Hanahan, that tries to distill the vast complexity of cancer into a set of underlying principles used to focus not only on specific lines of investigation, but on how biological programs interact to produce disease. Models such as this, once very daunting, are becoming more and more attractive as we acquire the computational capabilities that allow us to identify relationships between complex biological variables, pathway interactions, and to predict new models of disease that can be targeted to achieve prevention or treatment. Next slide. And an even higher level view is shown here. This slide just lists some of the biological programs that humans require for growth, development, and maintenance of health. We develop from a single cell. Our biology constantly changes as we transition through various life stages: child, adolescent, adult, older adult. And to keep us healthy in the face of demands from our environment and lifestyle, we have to be able to renew epithelium that provides a barrier to external insults, heal wounds, especially of complex tissues, respond to infectious agents, maintain the integrity of our DNA, function in the face of fluctuating nutritional inputs, and detoxify harmful substances, to name just a few of the incredible things that our bodies do every day. And when any of these critical functions are thrown out of balance, disease results. This heuristic reminds us, in a fairly simple way, of the value of fundamental research as it addresses the underlying principles that span multiple organ systems and multiple areas of disease. Next slide. At NIH, we're accountable to the American people. Our mission is to seek fundamental knowledge about the nature and behavior of living systems and to apply that knowledge to enhance health, lengthen life, and reduce illness and disability. We're called upon to justify how we spend taxpayer dollars to achieve our mission. And understandably, this accountability to the American public often means we are asked how and when we will make more progress for specific diseases. In addition, we often receive our funding from Congress with a requirement for studying specific diseases or conditions or areas of research. We also, however, know that the basis of our progress comes from curiosity-driven science. It may be many years before a strand of fundamental knowledge, which might at first seem to interest only a small number of academic experts, becomes critical for developing a new therapeutic agent or some other application. It is impossible to predict where important discoveries or insights will take place, or who will be involved in making them. Believe me, we've tried. You can't predict. Wonderful things come from unexpected areas. Therefore, supporting a very broad and diverse scientific portfolio maximizes the chances for breakthroughs. Demonstrating these principles, we can rely on the work of thousands of scientists at thousands of organizations over many decades. And I'll just say it again, we're very grateful to FASEB for your work to champion these areas of fundamental science, promoting both the workforce required as well as the work itself. Next slide, please. NIH must support research that encompasses the laboratory and the clinic. But we also increasingly understand that our work does not stop at the door to the doctor's office, but must extend deeply into the communities we serve. As just one example, we currently have a renewed emphasis on improving the health of the American people by addressing social determinants of health and environmental justice. To do this, we must do both sides of the equation. For example, eliminate disparities in health outcomes by studying healthcare delivery to find a way for those who lack access to clinical care to receive it. But we must also pursue technological innovations that will address how environmental factors present in specific communities, such as toxic exposures or regional effects of climate change, affect our cells and tissues in ways that influence health. So we really must encompass the laboratory, the clinic, and also the community in all we do. Next slide. So treatment success requires a fundamental understanding of health and disease. And now I'd like to use our response to COVID-19 pandemic as an example of where our investments in basic science have paid off, but also fairly glaringly where we need to do so much more. First, a success story. I'm sure you all know this. I'll go through briefly. When SARS-CoV-2 arrived in 2019, the study of mRNA and how it works to either activate or block protein production in cells had been studied for more than 60 years. In the early 2000s, NIH scientists identified the structure of a protein on the surface of the human immunodeficiency virus that allows it to enter human cells. This provided the foundation for considering structure-based vaccine designs. In 2005, a laboratory breakthrough showed that modified mRNA can safely deliver instructions to cells without overactivating the body's immune response. In 2015 to 16, cryo-EM was applied to capture the first three-dimensional images of the spike protein on the SARS-CoV-1 virus, the original SARS virus. 1 year later, based on earlier studies of coronaviruses and RSV, scientists found a chemical combination that stabilized the coronavirus spike protein, allowing the immune system more time in the face of this spike protein to make antibodies against the virus. Then in January 2020, when Chinese scientists shared the first genetic sequence of SARS-CoV-2 with the NIH GenBank, research teams from NIH, academia, and industry quickly used all of the research advances listed here to develop a candidate vaccine to respond to the emerging COVID-19 pandemic. And just 11 months later, in December 2020, the FDA granted an emergency use authorization to 2 mRNA-based vaccines that reduce deaths and hospitalizations due to COVID-19. Tremendous success story illustrating the critical importance of fundamental science. Next slide. But we're not done with this new virus in so many ways. Within the first 5 months of COVID-19 in the US, those infected began describing persistent symptoms. The term long COVID first appeared in May 2020 in a social media post from a patient named Elsa Perego. In December 2020, just as the first COVID-19 vaccines were approved under an EUA, Congress allocated $1.15 billion to NIH to address long COVID. We did not then have a clear definition of this new disease at all. It's just a lot of reported symptoms from a lot of patients. It appeared to share many clinical features of other post-infectious syndromes, such as myalgic encephalomyelitis, chronic fatigue syndrome, but we had no way of clearly defining the condition with the rigor required to develop treatment approaches. Through its Recover initiative, NIH launched large-scale observational cohorts, the first task being just to define the disease. Today, these included both COVID-affected and COVID-naive people, critically important, and also all of the different, were rapidly initiated to capture the many different COVID variants. This turns out to be very important. Today, these cohorts include more than 30,000 people and have provided a working definition of long COVID based on severity scored clinical symptoms. We all admit this definition is too subjective to be optimal, but lacking objective diagnostic tests, it's the best we now have to allow researchers to track the prevalence of the disease and to initiate much needed clinical studies. Data emerging from across the globe, you can see here on the bottom right, on a timeline listed in May 2023, shows that COVID affects different people in different ways. This can change with the new arrival of new variants. It can persist in tissues in some people for years, has a highly variable clinical course. Clinical trials to treat long COVID began in July 2023. Still lacking a definite understanding of the underlying pathobiology, these studies test the efficacy of longer-term antiviral therapy based on the hypothesis, without definite proof, that long COVID is caused by persistent viral infection. And then also studies are also testing methods to improve cognitive function, found to be helpful in other post-infectious syndromes. Next slide. Data continue to accumulate, demonstrate the many ways that COVID-19 causes harm to different organs. New treatment trials have been started, testing promising symptom-directed interventions and continuing to collect annotated biospecimens. And in a few weeks, NIH, you see there on the bottom right, is convening the scientific community to take stock about what has been learned about the pathobiology of long COVID from longitudinal cohort studies and from what else has happened around the globe, and to identify the most promising interventions to move into a next phase of clinical trials. It is very clear that long COVID is a serious, now common illness. It's not going away anytime soon, causes great suffering, and it will join other poorly understood post-infectious syndromes. Next slide. And those working to prevent and treat long COVID are all acutely aware that we're unlikely to achieve successful treatment without much new research detailing the exact nature of the disease. We're more or less still working in the dark, looking at many pieces of the puzzle without understanding the whole. As with other viral post-infectious conditions that have been described for more than 100 years, we're hampered by something as basic as the inability to definitively identify live replication competent virus in specific human tissues. We need new diagnostics and a new understanding of basic pathobiology. Next slide. Although some common principles are beginning to merge based on a study of known mechanisms of disease, the heuristic is helping out here a bit. We're fortunate to have tissues from carefully clinically annotated recovered cases and a large clinical research infrastructure to draw upon for future studies. But long COVID remains a very important example of how ineffective we are when we lack the knowledge base and technological innovation provided by fundamental science that is directed at understanding the basic pathobiology of a disease. Next slide. The contrast between the rapid production of a successful COVID-19 vaccine made possible by decades of basic research and our inability to adequately understand and treat long COVID provides a clear teaching moment for all of us that illustrates the importance of fundamental research. Next slide. NIH prioritizes investigator-initiated basic research. At the current time, 51% of our budget is directed towards supporting basic research. Next slide. The National Institute of General Medical Sciences focuses primarily on the basic community. I know Dr. John Lorsch is joining us here today. Thank you, John. NIGMS funds basic research that increases our understanding of biological processes and lays the foundation for advances in disease diagnosis, treatment, and prevention. Its programs also support research in specific clinical areas that affect multiple organ systems. For example, anesthesiology, clinical pharmacology, the condition of sepsis. A significant portion of its activities are directed to training the next generation of scientists and developing research capacity throughout the country, not just localized in specific geographic locations. Next slide. Other NIH institutes and centers also support basic research, but these research programs largely focus on specific diseases, organ systems, or populations. I'll just remind you, you can see how with long COVID, we've got at least 10 organ systems in play. So obviously, we're not going to solve a problem like that with just the work of individual institutes and centers. Through activities of the NIH Common Fund, many cross-IC initiatives in basic science address important research needs. I'll just give you some examples of current projects supported by the Common Fund. Transformative high-resolution cryo-electron microscopy, the 4-dimensional nucleome, extracellular RNA communication, the Human Biomolecular Atlas Program, and the Somatic Cell Genome Editing Program, to name a few. Next slide. I'd like to also call your attention to some funding opportunities specifically designed to promote curiosity-driven fundamental research. These include the NIGMS Maximizing Investigators Research Awards, the R35s, which are longer and on average larger than other NIGMS R01s. The intention here is to allow researchers to focus on doing research instead of writing numerous awards, numerous grant applications. These awards are flexible. The research can change direction as new observations or clinical opportunities arrive, and they're more stable because they have higher renewal rates than that of other R01s to promote ambitious research. Examples of research underway under the MIRA program include developing easier chemical methods for making mRNAs and other nucleic acids, which could transfer how we manufacture these molecules for use as drugs or vaccines. Inventing new technology that allows molecules to be visualized inside cells with unprecedented precision by combining fluorescence microscopy with cryo-electron tomography. Repurposing insights from organic chemistry to modify the plastics used in medical devices, which will allow researchers to vary how quickly polymer stitches dissolve in the body or adjust the rate at which an implant can release a drug. And finally, last example, using artificial intelligence and data science techniques to model both healthy immune systems and multi-organ system diseases like sepsis, and using these data for drug discovery. Next, the NIH Director's Pioneer Award is part of the NIH Common Fund's High Risk, High Reward Research Program. This award is for scientists at all career stages. This is the DP1, with outstanding records of creativity to pursue pioneering approaches to major challenges. This grant, the DP1, awards $3.5 million for 5 years. The second one, the DP2, is the NIH Director's New Innovative Award, also part of the Common Fund High Risk, High Reward Research Program. It supports exceptionally creative early career scientists proposing innovative, high-impact projects. This award provides $1.5 million over 5 years. Next slide. NIGMS also ensures the vitality of the research enterprise by supporting the training of the next generation of scientists across career stages and transition points. A goal is to support training or student research experiences in every state and across all educational and career levels, pre-K to 12, community college, undergraduate, post-bac, graduate students, postdocs, and faculty. The Trans NIH Maximizing Opportunities for Scientific and Academic Independent Careers, easier known as MOSAIC, program also supports a cohort of early career independent investigators from diverse backgrounds. For example, individuals from underrepresented groups conducting research in an NIH mission area. FASEB, ASCB, and ASBMB are all NIH-funded mentoring centers for the MOSAIC program. So now to close, next slide. I just want to go over 2 additional guiding principles. The first guiding principle was we have the full scope of the lab, the clinic, and the community. Our next one is that our work is not finished when we deliver scientific discoveries. We do view the totality of what we do as meaning that our work is only finished when all people are living long and healthy lives. And to complement this, next slide, progress, we know, is accelerated when advanced scientific methods, such as new data analytics, are applied to data that include everyone, and when new discoveries are rapidly and equitably adopted in clinical care. So next slide, please. Thank you so much for supporting the critical scientific foundation, allowing us to apply these guidelines to improve the health of all people. And thank you again for allowing me to join you today. I look forward to your questions.
Thank you so much for that presentation. We really appreciate it. We have questions in our Q&A, and we also had some questions that we sent you in advance. I think you answered some of our big picture questions. But as you know, one of the concerns that I think many of us have has to do with how you balance the portfolio between disease-specific investigation, which you just talked about, and a more global approach to understanding an application of biological processes, which kind of form the basis of R01 types of grants.
Yeah, so I can tell you unequivocally that our priority, frankly, in all areas, is investigator-initiated research, usually the investigator-initiated R01. Although we clearly understand and are increasingly applying other efforts like the high-risk, high-reward program, et cetera, because we know the traditional R01s sometimes, don't achieve what we're trying to in capturing the innovation that we see out there. We also look at programs that operate in the venture space. We have programs there. So one of the ways we do this is we try to have a variety of mechanisms that support different activities, but again, focus on investigator-initiated work. Easily 50% of what we do is in that, roughly, is in that space, because we count on the community to tell us, to vote with their dedication to science, with what needs to be done. But then obviously we have to have some focused programs, because to be perfectly honest, that's what Congress tells us to do. So we do have to meet, as I said, the full spectrum and tackle many other issues. But I will say, even in those areas, the priority is always given to the investigator-initiated work. I think we try very diligently across all of the institutes and centers to maybe put out challenges. Here, long COVID's a challenge. Here, we need to understand new imaging techniques that will let us find a virus. Challenges out there, but then see what the community gives us back, rather than be too directive.
Thank you. I'm sure you're aware that there's been a lot of concern about the ACD working group on re-envisioning NIH-supported postdoctoral training. FASEB has, I think, been fairly vocal about how we feel about this, and we appreciate the opportunity to respond to a recently issued RFI. I think some of the questions that we have really come out of what the outcomes of this is going to be, right? So,
yep.
For instance, increases in postdoc salaries. I don't think anybody minds raising postdoc salaries, right? The question we have always is, well, how are we going to pay for that? So, as I'm sure you're aware, budgets on NIH grants are always personnel heavy and also tight. And having to suddenly raise a postdoc salary is difficult. And so I'm wondering if NIH is addressing kind of that balance between the raising of the salaries and how we deal with this, like on our R01s, for instance.
Yeah. So thank you for that. And we've been giving this a lot of thought, because I think we all agree we want to see postdocs better supported. We completely embrace all the recommendations of the working group. However, the dollars are fixed. And, this year was like the worst possible year to try to roll, or at least the worst year in a while, to try to roll something out. And, it's conservation of mass here. I don't have an answer. We have so many dollars. We have to spend them. I will tell you that we can't make these decisions alone. This is why we're very grateful for FASEB's input, incredibly grateful. The other thing I am doing is I am spending every opportunity I can with university presidents, you know, heads of big faculty programs, and saying, what do you, given the fixed amount of dollars, and I literally ask them this question: what do you want? Do you want bigger grants, which would be great? Or do you, and bigger grants means fewer grants, or do you want more grants and they stay small? You tell us, because we do not have enough information to understand which is better, right? It's the hiring institution, the academic environment, which calls on so much more beyond just what NIH provides to support the faculty, to support, you know, to support personnel and careers. And we see one part of the puzzle, which is just what NIH can give. So I will say that we're, you know, I personally am leaning pretty heavily on asking the employers, the institutions, the academic programs, for their advice. I can kind of tell you what I'm hearing, at least so far. I'm hearing, if the choice is fewer bigger grants or more grants that are not smaller, everybody said they want more grants that are smaller. That helps the mix work better for whatever they're dealing with. So I'll tell you right now, we hope for more robust funding. We especially hope to continue to increase our training programs, because obviously training programs, that's easy. You know, here you go. Anybody we can have on a training grant, it should, you know, brings better flexibility and you know, better definition of support to a particular group of trainees. But increasing the pie and continuing to work with our academic partners and FASEB is where we're at with this. No ready answer.
It's a hard problem. I think we all agree with that. I'm gonna go.
The last thing I'll say, I'm sorry, the last thing I'll say is
right.
we're also aware that one size doesn't fit all either, you know? So
Right.
we're also thinking, how can we provide flexibility? You know, do you apply for a modular grant or you go with a traditional? Well, people have kind of voted with their feet and said, we don't want the modular, you know? So this is how we learn, frankly. So we need to continue those kinds of interactions.
Of course, the modular grants have not increased in, I don't know how many years.
Exactly.
So that's essentially what the issue is we can't afford modular grants anymore, right?
Agreed.
Yeah.
Yep.
So I'm gonna jump over to the Q&A for this question. So we know what NIH has achieved and are grateful for new programs, but scientists young and old are fleeing the profession. What is being done to ensure that the best scientists and talent remain in academic science? And by fleeing the profession, meaning they're not choosing academic science, right? They're still using their PhDs, but it's in some other area.
Yeah, this is a very interesting dynamic, and I'll say that there are voices out there that say, hey, that's great. We're glad we're training all these bright people that are going to industry because we want them in industry.
Yep.
So I'm taking a big breath about this one too. I do think that NIH is fully committed to our academic institutions, to their stability, their growth, to continue to nurture them to the fullest extent possible. That's where our dollars are directed. You know, we tend to engage in industry in a partnership mode, not in the kind of model. You know, our support belongs with academia, and we will continue to do so. The other thing I'll say is that the single biggest concern we had with this year, which was, you know, we had a negative balance on the total this year. Our biggest concern was the new generation and whether they would abandon academia even faster. We saw this with the sequester. You know, we all know we lost a generation with the sequester. I will say that in addition to providing the other thing we know, it's one thing to say, well, we're going to give a bunch of, give preference for a bunch of junior investigators awards. We're also mindful that we don't want a bunch of junior and training awards without a robust, mature, maintained mentoring scientific community as well. So I think we're in the same boat with you all, trying to balance everything. And we're always open to listen to any suggestions concerning how we could do this better.
So there's another question in the Q&A that several people thumbed up. So when NIH interacts with Congress, what misconceptions come up that FASEB can help dispel? So we're active in our policy and dealing with Congress. So what can we help you with?
Oh, where do I begin? About the misconceptions. No, I'm joking. I think so first of all, as I hope my presentation made it clear, the societies that you represent are absolutely critical, and your mission is perfectly aligned with ours. So I mean, I can't think of anything that your representation as it is right now, I think is perfectly aligned with things that will really help us with our goals. You've seen what I've described here. We really need to preserve curiosity-driven fundamental science, even though it's nice to say, Congress wants an Alzheimer's program. Congress wants a women's health program. I mean, we're just naming the ones that have popped up for funding this year, to also make the point and remind that we need the robust core funding, or those programs won't succeed either. And so, I know that's one thing in particular that I think is very helpful for Congress to continue to hear, because they constantly have patient groups and other constituencies in there clamoring for individual diseases, frankly. It's really important that our professional societies take the longer infrastructure, training, and education view that helps us make sure the entire enterprise is truly robust.
There's several questions having to do with peer review and the peer review process in the Q&A. So I'm going to take a few of those. What can NIH do to restore confidence in the review process, i.e. ensure a high level of proficiency of peer reviews and safeguard against kind of moving targets and erratic evaluations, that notorious reviewer 3 in the review process? That was my added.
That was your added.
My little added again.
I'm sitting here as an NIH director who's been the victim of reviewer 3 for sure. I'm with you there. I can tell you that under our director of CSR, Noni Barnes, the entire peer review process has really undergone a big scrutiny in every corner of it. And I think Dr. Barnes has a lot of ideas and principles that she, educational programs, new approaches to convening study sections. Dr. Barnes really has an entire portfolio of, I think, really forward thinking and I believe ultimately really helpful input to provide for this, and maybe we could, arrange a discussion with Dr. Barnes or something like this, because I don't feel as competent as I should be and able to speak to it, other than to tell you that first of all, I think she's spectacular, and she really has dedicated time and gotten much outside expertise in taking a fresh look at the entire system.
Yeah, so I'll just throw out, there was another question, a little bit different, had to do with the fact that most successful grants are conservative in scope and risk averse, just because of the way we have to write them, the preliminary data that's required for a lot of them. And it's very difficult for study sections to actually discriminate between grants that are in like the 10 to 25 percentile range. And so another question is, you know, is there a better way of discriminating these grants? Or is there even a better model for funding other than this peer review system that we have? And maybe that's another question for the head of CSR.
Well,
but actually, maybe not. I mean, yeah.
So I described the Mirror Awards, the D1, DP1, DP2. We're trying some other approaches that don't do the, safe and easy groupthink of a study section and really push innovation. We're going to see. So far, I think those results have been very positive. Perhaps if that mechanism that approach truly appears to be successful, it could be expanded. I'll tell you the biggest thing that would help. It's this splitting hairs because the pay levels are the funding levels are so low. That to me, I mean, if you want something that could ease a lot of this angst, it is to be able to provide more robust funding levels. And I know we're all hoping to see that. But I know, the National Academies takes this on, bringing a lot of minds to think about it. I've read the very extensive Brookings report that came out just a year and a half ago, 2 years ago, about this subject. I know that ARPA-H was designed and intended to break this mold and do it a different way. They really have a different approach.
Very different.
Very, very different. It'll be very informative to see the results of that, and how that complements what we're doing. I think right now our focus is on making sure that study sections are free of conflict, that they have very scientifically qualified, open-minded people, that the team at NIH who manages and oversees them sticks with principles that really do promote really equitable and appropriate reviews. So we're focusing on making our current process the best it can possibly be, focusing on and hoping that our pay lines become more generous, and but we're open to other and experimenting with a few other approaches.
I'm going to go back to the questions that you've already seen. This one has to do with NIH finalizing policies to ensure free, immediate, and equitable access to federally funded research. What do you see as the biggest challenge to implementation, and what steps can be taken to mitigate these challenges? As you know, all of our societies and our journals are grappling with this, as all the sciences. So
I guess I just need a little more clarification. Are you talking about access to data or access to
federally funded research data?
Data, yeah. Well, I don't know if you saw the last ACD meeting or looked in on the last ACD meeting. We described new programs. We are taking a new look at the National Library of Medicine. We are recruiting a new director of the National Library of Medicine and envisioning, because we really believe that NIH needs to do much, much more to serve as a central facilitator. That's the key word, facilitator of data access and data use for biomedical research. We envision a federated system, meaning, data can be held in many places, but there has to be the ability for researchers seeking those data to know where they are, to know how to access them, to have access to the tools and infrastructure, to the tools and analytics required to study them. We can do more. It is a very big price tag item, as I'm sure you're aware. I've described this vision to our legislators because I really believe this is one of the most important things we can do right now to, supercharge the work that is going on. So far, it's received a good reception. But it's all a lot of talk until we actually really start doing it. And I'm really looking forward to bringing on board a visionary new NLM director who can really help develop and lead this new ecosystem for data use and sharing.
Thank you. So a bunch of us live in states that have been affected by limitations on programs fostering diversity, equity, accessibility, and inclusion, including me. And we are all concerned about what effect that might have on NIH programming that of course we all value.
So we have not changed our policies, procedures, approaches whatsoever based on the things that are happening within the states. What we have done is we're carefully tracking what is happening. Are we seeing less diversity of our applications? Are we seeing challenges with young investigators? Are we seeing challenges with particular programs, applications for particular programs? I can say that I don't think we've seen any objective evidence that some places are doing worse, than others. But number one, it's too soon to tell. I think it's too soon to tell. We do not have enough visibility to understand if some of these programs are being negatively impacted. I'll say again, though, that our approach is not incompatible with what a lot of the states are saying. According to federal print law, we cannot select funding specifically on the base of race or gender, or, that's allowed to do that. But we are allowed to promote diversity of thought, diversity of geographic location, diversity of background, which can take into account someone's ethnicity or gender. So we can't award anything based on those principles. So it's a bit of a fine line, and it sounds like a bit of doublespeak, but we have not changed our principles at all based on what the states have done, nor have we had to. I don't know. We have, I don't know if Dr. Tabak joined us. Is Larry here?
Yes, I am, Monica.
Larry, you want to weigh in on this one too? It's so important. And I just want to make sure I've gotten it right, because I know you've been really helping me lead our understanding of much of this.
So thanks, Monica. No, I think you got it just right. There are certain principles we have to follow. You have to follow your state laws.
Mm-hmm.
We can't tell you to violate your state laws. In those instances where the state law conflicts with federal policy or law, then there's a conversation that has to be held with the program officer, because there are instances where you can do some, modification of specific aims, and sort of thread the needle, as it were. All of us, I'm sure everybody on this call, I know many of you, all of us, subscribe to the notion that diversity of thought is crucial, that we want to get the best talent from everywhere, across the nation. And so we have to keep, working at this. It makes it a bit more tortuous in some instances, but so far we have not, for example, had to have an institution withdraw an application, to my knowledge. If anybody out there has an example of that, I'd certainly be interested in knowing. John Lorsch, you may also want to comment on this, because I know some of these things directly intersect with your equities.
I don't have anything to add, other than, as Larry and Monica said, I would, urge you to speak or urge your faculty to speak to your general counsels. We do get a lot of requests for legal advice, which we can't give you. And so I think that would be something important for you to bring back, that you should be talking to your institution's legal counsels.
I'm just
Thank
going to
you.
reiterate what Larry said, because one mistake I do see that we really, really don't want to have happen is people don't reach out. So, if there's a question, if you think there's a problem, reach out, because there are, many people within the programs who are all too willing to try to help thread this needle, as Larry said. And it changes every day, right? It seems to change every month. So we're going to have to be able to move with this.
Thank you. All right, I'm going to move to a question that has to do with shared resources and core labs and facilities. Would NIH consider conducting a census of the shared resource and core labs and facilities? They've become essential for NIH-funded biomedical research conducted at R1 and R2 institutions, providing expertise and access to advanced technologies. Having an accurate count of these shared resources would perhaps enable institutional sharing of the resources. So that's one question on that.
So let me ask John to address this one, because I know this is like right in what John thinks about and tries to promote all the time. So John, you take this.
Thank you. So actually that was attempted about 10 years ago, I would say. Larry may remember exactly. Jim Anderson's group tried to do this exact thing that's being suggested in NIH, make a census of core facilities, and it proved to be extremely difficult to do. Partly because, to be frank, partly because institutions didn't want to give us, in many cases, a census of the core facilities. And when Jim started to do a survey online, just trying to look in websites and find what was there, it appeared that there was quite a lot of duplication of core facilities, which might have been a reason they didn't want to, give us a census. So that may be the answer to the question, that is an issue that needs to be addressed. I know FASEB has been thinking a lot about core facilities. Our approach, as I've told many of you before, is to try to take it a level up and support national and regional resources to get better economies of scale and get more democratization. And so I think that's the approach that at least NIGMS is taking to that.
Got 2 minutes. I'm going to give you one more question. FASEB continues to be a strong advocate for initiatives such as the IDEA program to foster research capacity and workforce development in areas with historically low NIH funding. In addition to existing efforts such as COBRE and NBRE and CTRs, et cetera, what additional programming would NIH like to offer to continue stimulating research opportunities in these regions?
So one of the things that we've discussed, this is much more in the clinical realm than it is in the fundamental science realm, but there's no reason why, if there weren't similar things, we couldn't adopt a similar approach. So obvious. We looked across all of the institutes and centers, and we have NCATs, and we have CTSAs, and you can note some infrastructure duplication when it comes to translational and clinical research. And that, on top of that, a need to provide much more support and infrastructure for places like the IDEA states, et cetera. So one of the things we've done, first of all, is through a program called Care for Health, develop a clinical research infrastructure with participation from CTSAs and some of the IDEA CTN programs and PCORI and PCORNet and many programs out there, and unified them and brought them into a single mechanism that can be used by any of the ICs of NIH. Last of all, this involves the now the primary care environment, a focused environment that, really needs help, needs more support to be able to conduct research. So I know that's not exactly what you're looking for, but similar ideas would be most welcome. Because we have 27 institutes and centers. We can always think of a way to collaborate better. And I will say that one of the real pleasant and most welcome surprises, frankly, after having been the NCI director, and then coming over to this post as NIH director for both of those years and posts, just the willingness across all the institutes and centers to collaborate has been truly amazing. So there's a big willingness to do that if it makes sense to serve the science. So we're all ears.
All right, so we will feel free to make sure that we continue to provide you with our ideas and information. So thank you so much for coming and meeting with us. We appreciate the partnership that we have with you, and we look forward to continuing that.